The overall goal of this experiment is to pre-concentrate bioagents by confining the ion concentration polarization zone between two identical ion exchange membranes.
This method can help answer key questions in the biosensing field, such as how to detect a low abundance biomolecule when its concentration is less than the sensor's limit of detection. The main advantage of this technique is that we can generate ICP zone and pre-concentrated bioagent in a very specific region, regardless of operation conditions.
To begin this procedure, use conventional photolithography or deep reactive ion etching to fabricate silicon masters for the polydimethylsiloxane microchannel and cation selective membrane molds. Place the silicon masters in a vacuum desiccator with about 30 microliters of trichlorosilane. Close the desiccator and silanize the silicon masters for 30 minutes. Next, mix together a silicone elastomer base and a curing agent in a 10:1 by weight ratio to obtain uncured PDMS. Degas the mixture under vacuum for 30 minutes. Then pour the degassed uncured PDMS onto the silicon masters. Remove bubbles from the PDMS with a handheld blower, and then cure the PDMS molds at 80 degrees Celsius for 2 hours. Separate the cured PDMS components from the silicon masters. Use a knife to shape each component into a rectangle. Next, cut 2 lines in the cation selective membrane mold from the edge of the mold.